Uko Fukami and Professor Dr. Ibrahim Taltawi, the organizers of this conference, to give me the opportunity to participate in this conference. Also, deep thanks to all participants in this conference. Welcome. And good morning, good afternoon for others. Today I will give lecture about new strategies for treatment of COVID-19 and evolution of SARS-CoV-2 according to biodiversity and evolution theory. Outlines of my presentation: structure of virus, coronavirus structure.
Sorry, you didn't share your screen.
I share my screen now.
No, I still see you. Press the green share icon, share screen at your Zoom. Yes. No, share the PowerPoint. Choose your PowerPoint.
Yes. Something appear or not?
Yes. You can enlarge for slides.
Yes. You see outlines of presentation.
Yeah, but one slide. I need you to make
yes,
it.
Yes, you now start by slide. Yes.
Okay, in full screen.
Excuse me.
بص يا دكتور صبحي، خليها تشتغل كانها برزنتيشن عاديه، يعني افتحها شغلها، هي كده عندي باينه انها برزنتيشن مش شغاله. حضرتك بس تدوس على السلايد انها تشتغل تشتغل.
طب اشتغل كده حضرتك، اشتغل كده ساعتك.
انا شايفاها هي مش شغالين، هيشتغل واحده واحده. بص حضرتك يا دكتور صبحي، نشتغل على الباوربوينت. عندك هتلاقي ايوه شايف اللي هو الكاب، علامه الكاب.
تمام الكاب، تمام ساعتك.
اللي جنبها يا فندم، اللي جنبها يا دكتور صبحي. لا، الناحيه الشمال، اخر واحده.
اوكي،
اخر واحده خالص. ايوه دوس عليها.
دوس على ساعتك كده.
ايوه بس كده. شكرا دكتور صبحي، ثانك يو.
واضحه؟
ايوه
واضحه جدا. I will start from my presentation: structure of viruses, coronavirus structure, principles of COVID-19 treatment through microRNA treatment, VH and COVID treatment, and evolution of SARS-CoV-2. Now we start the story from the first. God created everything, created protein, nucleic acids, lipids, carbon, and everything. So we see now this is a protein. And in the next picture, we see here protein and nucleic acids. The interaction between nucleic acid and the protein results to viruses. So the virus, I consider the virus is the nucleus of all organisms. Why? Because any nucleus contains the nucleic acid and the protein, or composed of nucleoprotein. So the nucleoprotein, this is a nucleoprotein, nucleic acid and the protein. This is like the nucleus of all organisms. So I consider the virus is the nucleus of life, nucleus of all everything. This is the very step. The second step for evolution of the viruses envelope. Now we know nucleoprotein, nucleic acids and the protein. And some development takes place for the viruses where envelope was formed. This is envelope of the viruses. This is specific more for the animal viruses. When I call or mention the viruses of the virus is the nucleus of the life, if we imagine it, the envelope and the nucleoprotein separated and give space, this space will be formed cytoplasm. So the life, we know the unit of all organisms is the cell. So this gives some imagination or the relation between the viruses and cells. What is the SARS virus? Many colleagues show the what is the SARS. SARS is the severe acute respiratory symptoms, and MARS. This is the COVID-19 arise from this viruses. The history of viruses, HCO COVID-29 and HCOV, these all viruses, the COVID-19 arises also from this is viruses. So, how we treated the COVID-19? We have principle to attack SARS-CoV-2. We should use the same language that viruses use it to attack us. So the SARS virus composed of ribonucleotides. It is RNA viruses with strand that has nitrogen bases, adenine, uracil, guanine, and cytosine. So SARS-CoV attacked its host through its genetic materials, which is RNA. Therefore, genetic antigen must be designed like microRNA against the genetic material of the SARS-CoV-2 in order to get rid of it. This is the main principle of how to treat the COVID-19. Strategy of treatment, to treat COVID strategy was assumed through a design, a complementary microRNA to 3 UTR of the viruses. So when combined or this is combined with the virus at 3 UTR, this will be inhibit the translation of viruses and also get rid of degradation of the virus. Here, the picture of the viruses. We focus here the nucleic acids or the RNA here in this is, and start by 5 cap A and 3 poly A, 3' poly A. This is exactly like any messenger RNA. Any messenger RNA composed of 5 cap and end with 3 polyadenine. This is the main structure of those. And we see here is receptors, the envelope composed of receptors, and also agglutinin receptor. And the virus is dangerous where the hematoagglutinin this protein coagulate the RBCs and form a clots, blood clots, and this is the dangerous of the virus in this area. What is the microRNA? MicroRNA produces transcripts from polycyclic gene forming pre-microRNA, which proceed and many steps like this is the steps here. So its original is double strand microRNA, and then it gives 2 strands here and separates and gives microRNA, and the microRNA attached with 3 to R for inhibit the translation or cleavage A messenger RNA. Also, this is the same picture like before. Here we see here that any biological process start from DNA and they give RNA and protein. Here when microRNA attached with messenger RNA, here it prevents the translation for formation of protein. So this is photo is very nice. We see here microRNA attached with a complex protein, and when we bind with target messenger RNA, it will lead to degradation or inhibit the translation of or block translation processes. And this is our aim. Our aim here to block the the SARS-CoV-2 or degrade it through microRNA, and this is also the same. This DNA and they give messenger RNA and they give proteins. This is a central dogma of molecular biology. And here microRNA, when microRNA attached with messenger RNA, it will block translation processes. Antiviral, recently they found some microRNA act as antiviral defense. So here we list this, some microRNA act as antisense for the virus or antiviral activity. Here we see this, a good photo here. When virus attack cells and the above cells have microRNA or antiviral microRNA, it can bind with the genome of the virus and degrade it, and we see here no virus or no replication. In the second cell here, the second cell not has antiviral microRNA, so here we see the viruses enter the cells and propagate it and replication and so on. A new mode of action of plants that used for coronavirus. We hear to Dr. Fatima, and she speak about the dietary compounds and so. So some plants maybe contain microRNA or antiviral, maybe contain antiviral microRNA that can protect us from the COVID-19 or many viruses. Also, next second pathway for the plants. Also, it may be we will see in pH treatment. So this is new strategies plant we put here. New strategy for protective effect. Plants through containing microRNA beside antioxidant. All over the world, in a plant, antioxidant and protected antioxidant or increase the immunity. But here in this presentation, we add new strategy for protection effect of plants through containing microRNA as antiviral or protection from medicines. This is so easy. MicroRNA enters easily to mammalian circulation naturally, and then combined with high density protein and enter the cell and cause gene regulation or for the cells. Mode of action and delivery of microRNA. This is how we can use microRNA for treatment or the delivery. So if we sense microRNA antiviral, anti-COVID, so this is will be encapsulated with exosomes, and we all know the importance of exosomes can enter the cells easily, and we can go to the target virus or target gene easily here. So we can see now plants and foods also contains antioxidant, as Dr. Fatima saw that, as well as microRNA, which play an important role as an antivirus. Treatment of COVID-19 by plasma of recovery patient. Some methods, our recent methods appear. We can take plasma from recovered patients from COVID-19 and injected the patient, and this is give a good result. We now explain what is the mechanism. The mechanism of this is any virus attack the body will attack him with anti-microRNA or antiviral DNA. So when we take a plasma from a recovery person, this will contain antibodies, firstly, this is well known, and besides this we add, it may be also contain microRNA that protect the patient from COVID-19. This is well known, this presentation, the microRNA, two types, dietary and non-dietary. Non-dietary, this is produced by the cells, and dietary produced by the plants. Coronavirus induced production. This is we approve this avian infection, bronchivirus, this coronavirus. When the coronavirus infected the avian overexpression of GGA microRNA was observed. This is overexpression of gene. This is inhibited IB virus replication. So this is explain why the microRNA can be good for protection of us from the viruses. Host response to viral infection. Several microRNA have recently emerged an important regulation of host response to viral infection, a critical role of class small RNA and the HSA micro 1975 in hosts defense against influenza viruses. So we concluded that the very thing, microRNA could be used as a new strategy or a new treatment for COVID-19 through design a complementary design for the 3TR or 5TR ORF. So this is this microRNA with high density lipoprotein and injected the patient. This will be reach to the viruses and interact with it, and could be inhibited the replication or inhibit the translation, and also can be degraded the virus. This is the conclusion, previous conclusion from the part. The second strategy is the pH. pH play an important role in all biological processes, receptor binding domains. We see previously we see the virus has spike. The spike is a glycoprotein, and this is the glycoprotein. This is a protein for attachment to the receptors of the host, and we will know that any the domain connection with other needs certain pH. They was found in influenza and some coronavirus. When pH is acidic, this is help fusion of viruses or to enter the viruses to the cell. So if we raise the pH to be alkaline, and so this we can be the conjugation between the spike or glycoprotein and the receptor will be weak or interrupted as here alkaline pH here, the fusion and of viruses were interrupted. So we can protect us from the first step for the entrance of virus to our cells through keep the pH of our body as an alkaline cell. And it is well known pH of blood is 7.4. So this is slightly alkaline. So we must be keep to this alkalinity of the pH of blood and the body. This is will be through the dietary. So Dr. Fatima showed the importance of some plants, and so for COVID-19, we add a new strategy where the some plants like lemon juice and so and vegetables and so maybe keep the alkalinity of the pH of the our body, so it will be protected us from the viruses or from SARS viruses. This is we will see here the COVID SARS here, how the cell fusion and enter the cells. Here we see here, this is the coronavirus, and this is glycoprotein, and this is the receptor on the cells, and this is will be conjugation between our attachment between these receptors and between the glycoprotein and the receptors, and the pH will be cut. This is steps here. When acidic protein, it helps for virus fusion to the cells. When we alkalinity, it will be prevent or inhibit the fusion of the virus into the cells. This is the glycoprotein of the viruses. This is show the glycoprotein of the virus and the receptors of the cells. We hear, all of us hear about hydroxychloroquine and pH. And many people talk about it, and the hydroxychloroquine is very, very important for treatment of COVID-19. What is the mechanism of hydroxychloroquine? When enters the body, hydroxychloroquine, and it will be intercellular pH rise and inhibit a series of viral replications and recombination. So the mode of action when we raise the pH of our body, will be prevent or inhibit the fusion of the virus to cell membrane to entrance to the cell, or go from inside to out to attack another cells. Here we see here, this is a coronavirus fusion, and this is here hydroxychloroquine. This is made alkalization here. So we focus to increase the alkalinity of our body, not so high, but about 7.4 till 8 in some critical, because the high alkalinity cause many problems of our body or health problem. Evolution of the SARS here, we will know how evolution. We will hear in the future about the SARS-CoV-2. It will be developed daily. Why developed virus SARS-CoV-2? How developed? When viruses or intimate contact between viruses to each other, or between viruses and bacteria, or between viruses and the host in living or in living system, because viruses, our body is a non-living organism, but it must be inside living organism. So the intimate contact of virus with bacteria, fungi, or host, so horizontal gene transfer takes place between each other and the virus evolution. So we will hear about many SARS-CoV-19 will be appear in the future through the intimate contact between the viruses and another organisms and so on. Here we see here, this is bacterial transformation. Here we see it, the major bacteria can transfer from bacteria to bacteria. Here we see here the bacteriophage also inject, and this can interact with the bacteria and so on. Here we see here between bacteria and bacteria, horizontal gene transfer takes place. So through horizontal gene transfer or microRNA transfer, we will see and we will hear about large evolution for the virus in the future. This is a horizontal gene transfer. So mutation in SARS-CoV-19. A recent study shows that 42 missense mutations were identified in all the major non-structural and structural protein of SARS-CoV, except the envelope protein. 29 missense mutations were in the ORF, opening radical frame 1AB, polyprotein. The important, 8 in the spike service glycoprotein. So mutation in the spike service glycoprotein might induce its conformation changes. So it cannot be connected with or inhibit the diffusion fusion of the virus to enter the cells. Here we see opening radical frame and spike and mutation, and we see here many mutations that takes place in viruses. Here, not all mutation is dangerous, but some mutation is benefit. So when a mutation takes place for the spike or for the glycoprotein, this will be inhibited. This is glycoprotein to bind with its receptor and interrupted it, and this will be benefit. Fortunately, mutation in the genome regions of the virus spike may weaken the virus's attachment to its receptors in the host cell. This may explain the extent of the variation in the severity of the response between people each other and between different countries of this disease. There are future fearing. Yeah, in this picture, we put future fearing maybe takes place. We expected it. What is the future fearing? About the COVID-19 or SARS-CoV-2 transform to retrovirus. This is a big problem. If the SARS-CoV-19 transformed to retrovirus, this will be converted to cDNA and DNA and become part of our genome. This is future fearing. But this is will be some study or study all over that should be done about the recovery people, reinfected people. Somebody is infected and recovery and infected again. So we will see this is the retrovirus. If this is a simple experiment where we take the sample of the patient and do not put reverse transcriptase to cDNA. If this samples give positive result, so this is indicates that the virus is converted to retroviruses. Here we see some viruses, retroviruses, had reverse transcriptase. So we can see here some all over the world, or the World Health Organizer said that keep distance between individuals. Why? This is recent work for me. That the DNA can transfer from person to person. Here, good picture. Here we see the first one is a person, the third one another person. So when we shake hand and we take the fingerprint of one of them, we see here peaks of the another present in the hand of the another person. So the cheek hand, this is could be transfer the viruses and could transfer DNA from person to person. So we can conclude our work. We can protect ourselves, or we can attack, or we can treat the COVID-19 through design a microRNA for 3U2R of viruses and put this is in high density protein, and this will be bind to the virus and it prevent to replication of viruses and prevent to translation of viruses, and also maybe degrade the virus. So we can get rid of the viruses through microRNA. The second strategy is the pH. So when to keep our pH is normally 7.4, this is very, very good. And this will be takes place through some dietary, some alkaline foods, and so on. And also the evolution of viruses. We expected daily that viruses will be new species of the virus will appear through intimate contact of the viruses with bacteria and organisms, and through horizontal gene transfer. And we have future fearing from the virus to transform to retrovirus. Thank you for all.
Thank you, Dr. Sobhy. It's a very important picture. So I would like to welcome any question for Dr. Sobhy.
So share? Yes.
Yes. Any questions?
Any question in
Yes,
micro?
your specialty is very unique, and I think is pure science. So no one can get a question. You are the expert here. So
So the idea, I published this is idea in journal. After 6 days, I found Omina company produced treatment for COVID-19 by microRNA, and called this is recipe COVID-19 treatment and so. But alhamdulillah, after me with 6 days, published.
you know, it's very important to register any clinical trial or any trial on international websites for trials, so you can preserve your idea. Yes.
Yes,
And,
I hope that.
you know, many scientists know in right way how can preserve the property and intellectual property, and what about copyrights issue. So if we I did a course in WIPO, World Academy of Intellectual Property, and the first step, if you have an idea and
Yes.
you think it's a very important
idea design application.
before design and application, you can sign this idea.
Yes, yes.
In office, I think here in Egypt there is an office in scientific academy.
To best of my knowledge, this is the first idea appear for treatment of COVID-19 through microRNA.
So why you didn't register it?
To best of my knowledge,
No, I mean, if you have make sure that you is a first idea, why you didn't sign it, or you register your idea to have a
I published it in journal, and I will be send this is the publication to you, and
you reserve your rights by publication. But I mean,
yes.
Before publication, there is another step to sign before publication. In signing process, you can register your idea
Yes.
with a serial number of copyrights. So anyone can take it, and you can work free.
Yes, and my presentation, you can send my presentation to all participants.
Thank you so much.
Today.
Thank you.
I sent it to you yesterday, yes. And you can send it to all participants to read it.
Thank you for your kindness. Thank you for your kindness. So you spread your knowledge for all. This is a very good.
And also my paper, or the link of my paper, you can send all to all participants also.
Okay, I will make it public for all of us, and I will put it in our Facebook group, as well as I will put it in chat room after we finish. So I didn't see anyone write a question to Dr. Sobhy. So
any questions?
And yes, Dr. Ibrahim, and didn't hear Dr. Ibrahim is an expert and can give you some questions.
Any questions? Any questions?
Any questions? So
sure.
maybe after presentation is over, they will think about questions.
Okay, no problem.
Thank you to Dr. Professor and Dr. Sobhy, Professor Dr. Sobhy, for joining us today, and for all participants for their patience and their keeping with us all these from nearly 2 past hours. I would like to present our tomorrow lectures. Tomorrow we'll start at 12 p.m. also. We have 4 lectures tomorrow, inshallah. I hear. Okay.
Some Japan from Japan need to talk, or
Okay. I will read from my phone because I couldn't share. So, Professor Fukami, do you want to say some words in closing? Professor Fukami, you are here? Professor Fukami?
Okay.
Do you hear me?
Yeah.
Yes. Good afternoon, my dear professor. So, would you please give some word in closing till I found the presentation for tomorrow? Please.
Thanks so much, all the participants and also the lecturers. We are very happy to hear the new information and new research, and also some special field. So I suggest that we want to publish the collection of papers supported by JSPS. There are 20 lectures, so that the lectures, if you agree, all the lectures agree, please send me the papers, and we publish online and also the real book. How do you think?
Very nice, so nice. I am agree. It's a very good opportunity to all of us to have a paper published by the SPS. And so we can ask participants and speakers to send the abstract, right? Abstract for
the abstract or papers. There's some
paper. You need paper.
Yeah. If all the people want to make the abstract or papers, perhaps for the researchers, the paper is better, I think.
Okay. So I see.
Your side.
It's an excellent idea. So Dr. Sobhy, you can send any part of your research to publish in JSPS, and I will send my paper also to publish. I have paper for presentation in the third day, so I will send you my abstract for the paper. Dr. Itri and Professor Dr. Itri and Professor Dr. Fatima can also send paper for publishing. You hear me, Dr. Itri?
Yes, ma'am.
Dr. Fatima also, you hear me? So,
ma'am.
Okay, it would be great opportunity to publish paper in with JSPS International. So, do you mind to join us, or it's okay?
Yeah, ma'am. May I have a question?
Sure.
Yeah, ma'am. Do we have to send the abstract by today?
Okay.
I mean, can we have some more time, like 2 or 3 days or so?
Yeah, it's okay.
2
Perhaps
days.
no. End of September or October, we collect the papers and we will publish it. Is it okay?
Yeah, ma'am. Yeah.
Very good. This time we have nearly 1.5 months or 2 months, maybe 2 months to send abstracts. It's really be good time to write and to edit any abstract or paper, right?
Yeah. And we have to make how to write the papers or something, and how many words, and how to make the chapter or something.
Okay. You will send them template for abstract, right? The template or the way we all uniformly write.
Actually, I have no abstract now. Your abstract, perhaps Walaa Saad has.
Okay. I mean the way you want to all of us write the same abstract in one format, right? In one form. A number,
I'm
number of abstract word, number, the style.
I'm wondering the paper is better or abstract is better. So please hear the opinion from the lecturers. Today, not so many the lecturers are joined, but perhaps from your side, CC mail or something.
Yes, I
Please
have
ask.
all mails.
Yeah,
Okay, I have
please.
all mails, the mail of all speakers, as well as I have the mail of some registered participants. So I will propagate the idea, or propagate the request for sharing abstract or full paper to be published on JSPS, right?
Yeah, please.
Okay, thank you so much.
My word.
It's a very,
Thanks so much
very good news.
to participant and also the give the lectures. Thanks so much. See you tomorrow. Yeah, and perhaps the Professor Tantawi is not here.
Yeah. I will put here.
Rahim.
Just a minute. Dr. Raged, are you here? Dr. Raged?
No.
No.
Perhaps.
Professor Dr. Ibrahim, not here. It's okay. I will share the agenda for tomorrow. Just a minute, please.
Professor Hisham, you want to raise a hand?
Yes.
Please.
Yes, I just a very short note. Of course, you are doing considerable effort, and it is excellent idea. But if you please kindly could keep track of time, this would be really highly appreciated, because we arrange our schedule here in Sweden with the lab and with other duties. So if you can keep track of the time, would be great.
Yeah. We will negotiate by CC mail or something.
Yes. Thank you.
So make the closing, Dr. Walaa.
Okay, for tomorrow, third day of our symposium, we have 4 lectures. Professor Dr. Shumari from Sister Nivedita University from Calcutta, India. She is a director of Global Affairs, International Relations and Outreach. And she will give a talk about COVID-19 and the need value based transformative and authentic leadership. The second speaker will be Professor Dr. Rakesh from also India, but another university. He's an expert in statistics. He is assistant professor at part of Mathematics and Statistics University of SCHEM. And we will give lectures about change rules of statistics in COVID-19 pandemics. It will be so interesting lecture because all of us publish many paper, and in COVID-19 issue, it will be so important to know the role of statistics in this crisis. Also, we have a third lecture from Professor Dr. Raffaello Selegeto from Brazil. She is a Professor Doctor in Federal University of Minas Gerais, Brazil. She is an expert professor and will give her talk about the regional design of MP Pro inhibitor to treat the COVID-19. And we have the fourth lecture, but Professor Dr. Ashraf Abedi, but he has some problem in connecting us tomorrow. So this lecture will be replaced by another one, and I will send the topics and the hello, yes, by tomorrow or by late tonight. So I will be happy to have all of us tomorrow. We will start sharp by 12 p.m. time, and by 10 a.m. by UTC Universal Time Zone. And all our lectures will be recorded, and today by 9 or by 8 p.m. we will publish all recorded video on our YouTube channel, and will propagate the YouTube link for its for this lecture to be permanent and forever on the YouTube channel. So don't worry, you will have all lecture video recorded and stay forever on YouTube. So I would like to say goodbye now, and I'm pleased to have all of you with us today. Thank you, Dr. Professor Sobhy. Thank you, Dr. Fatima. Thank you, Dr. Italy. Thank you, Dr. Hisham, and thank you, Dr. Amr, Professor Dr. Chutub for is joining us. Dr. Hani and all of participants, Dr. Nermeen, Dr. Reda, Dr. Nili. So I'm so happy to have all of you today with us. Stay tomorrow, and I will share. I will send an email for the tomorrow lectures and for today link for YouTube. Thank you.
دكتور ولاء بعد اذنك، السلام عليكم.
عليكم السلام. مين؟
انا سعيده جدا بالمؤتمر الجميل ده، والعنوان بالذات especially انا السستينبل ديفلوبمنت في الكوفيد كمان في النقطه دي.
Excuse me.
It is very new, it is unique, it is novel to address this issue, and I'm very thankful that you allowed me to be with you with Zoom.
Thank you, Dr. Reda. Can you introduce yourself, please?
I'm Professor Dr. Reda Al-Badawi, Professor of Hepatology Gastroenterology, Bana University, and I have interesting lecture. If you have time tomorrow, can I present it? Related to comorbidities and liver disease and the COVID. Sorry.
Yes, Dr. Muhammad Salah.
Dr. Muhammad Salah.
mean tomorrow for presentation tomorrow?
Just cover it.
If you have a time, it is ready too.
It's okay. I will check with you. I will check with you.
Thank you.
In phone call. So thank you so much. And
you're most welcome. Thank you very much for the fantastic seminar going on.
Yes, fantastic, really. Yes.
Hi, Doctor. Hi, Doctor. How are you?
Yeah, I'm fine. I'm just listening to all the lectures.
Wow, it's my pleasure to have you today with us, and it's a very great pleasure.
Two of my colleagues are here, and also there are so many others. So naturally, I should be here.
Yes, I am so happy. Dr. Fatima and Dr. Tia joined us by very amazing lectures, very unique.
It's a very good idea by the Fukami regarding this JSPS publication of the proceedings of this particular seminar. All right. So I think the template followed by around month of October, if you can get this particular publication proceedings out, it should not be considered as a publication, but it should be considered as a proceedings of the seminar. Okay. All right, and it should be peer reviewed also.
Okay, dear Professor.
I think, thank you so much, the Professor Chaturapati.
Yes.
Yeah. I think the proceedings is perhaps if we make the proceedings, it needs 20 persons. But if someone wants to publish how to say, the papers, also JSPS support something, can support the wrong one. So, so that we will negotiate the 20 lecturers from Dr. Walaa. Perhaps she will mail all the persons. How is your opinions? It's better, I think.
Okay, yeah, but you know that should go through some peer reviewed, because peer review is very important to get the peer review for getting this quality of the publication. All right.
Okay, okay. So I will make something with my Tokyo office.
Exactly. Yeah, you should talk to your office, JSPS office, and I think that will strengthen the quality of the papers also.
Okay.
Yes, better to be international peer review application to be have a very good impact. Yes, all right.
I will seek the appropriate reviewer from our the society, science society.
Exactly. Yeah. You discuss with them, and I am sure that they will appreciate this particular command.
Also, if you know someone from India or Egypt or Brazil or UK, we accept.
Yeah, we should have. Yeah. All right.
Yeah.
Thank you very much for your suggestions.
Thank you so much. I am now reviewing international paper also from Indonesia. So about COVID-19 and immunization. So it will be a great opportunity to have peer-reviewed international reviewer to this application. Yes, you are right.
Thank you.
Thank you, Thank you, dear Professor. Thank you all.
All right.
Okay, thank you. So sad to say goodbye, and this is the time for leaving for us. I'm so happy. I know in India now it's 5 p.m. right?
Yes.
So enjoy evening and have a great time. Thank you, Dr. Sobhy, and all my dear participants and my dear speakers. I am so I can't express how I appreciate all your time and the effort to present today with us. And I wish all the upcoming 3 days will be so successful and happy for all of us. And thank you, Professor, for coming for this good news about publication.
Definitely.
It made me so happy. And I sure all of our speakers, it will be a great opportunity to publish because it's a very important issue to open our science from laboratory to all worldwide and global sharing. Thank you so much. Thank you. So goodbye.
Okay. Enjoy the rest of the day, and let's meet tomorrow. All right.
Okay.
Bye bye.
Okay. I will with you. I will with you, Professor, dear Professor. Goodbye.
Yes. Bye bye.
Bye bye.